So when looking at gene therapy, right, what we're supposed to be looking at here is generally what is gene therapy really about, right? And then looking at the success and challenges, right? Successes and challenges of gene therapy specifically. Here will be discussion, right? It's gonna be discussions, right? So I want you guys to speak up. I want you guys to say something, right, in relation to the information. Okay. All right. So let's discuss using an example. For example, cystic fibrosis is a genetic condition that affects the proteins in the body, right? People who have cystic fibrosis have a faulty protein that affects the cells, the body cells, its tissues, and the glands that make mucus and sweat. Normal mucus is slippery and protects the airways and digestive tract and other organs and tissues, right? So we have that there. Gene therapy, right, holds a great promise for the treatment of genetic disorders such as cystic fibrosis, but it faces significant challenges, right? What we're going to be doing is discussing, right, the challenges and successes of gene therapy on a whole, but keeping in mind the context of cystic fibrosis. So I'm using the context of cystic fibrosis to explain now the successes and the challenges of gene therapy. All right. Before we move on, what is gene therapy? Anyone? What is gene therapy?
The treatment of diseases by manipulating the genes in your body, sir.
Lovely. All right. So the treatment of diseases or illness, well, diseases, right? By manipulating the body's genome, genetic material, by using a gene, okay, to treat it. So let's look at this now. The challenges. Delivery of therapeutic genes. This is a challenge. So one of the major challenges in gene therapy is the effectively delivering therapeutic genes to target cells. In the case of cystic fibrosis, the defective gene responsible for the disorder is the cystic fibrosis transmembrane conductance regulator, the CFTR gene. Delivering a functional copy of this gene to the cells lining the airways poses challenges due to the complex structure and barriers of the respiratory tract. So one of the challenges, gene therapy we may have issues with delivering therapeutic genes to where we need to go. Notice all I'm doing is straight up answering the questions from the syllabus, right? So this is one of the challenges. All right. We have an immune response to know. Another challenge is the immune response elicited by the therapeutic vectors used in gene therapy. So viral vectors are commonly used for gene delivery, right? Therefore, if we implement viral vectors into the body, we're gonna have an immune response, right? So it will limit the effectiveness of the treatment and cause adverse effects overall, right? So in some cases, the immune response may recognize a viral vector as a threat and neutralize it before the therapeutic gene can be delivered to the target cells. So that's another challenge there, the immune response. All right. Regulation of gene expression, right? Achieving the appropriate level and duration of gene expression is crucial for the success of gene therapy. Regulating the expression of the therapeutic gene to ensure it is expressed at the right time and in the right amount without causing toxicity or other complications remains a challenge, right? So remember, if we express a gene too much and it's overexpressed or underexpressed, we may cause issues. So that's another issue. There are challenges in regulating gene expression. All right. So literally, I'm answering all the consequences, so all the challenges rather. So I'm giving you all the challenges, and this is the third challenge, right? The fourth challenge, right? Before we move to successes, right? We have the fourth challenge. All right. So gene therapy approaches must minimize off-target effects where the therapeutic gene is inserted into unintended locations in the genome, potentially leading to harmful consequences such as insertional mutagenesis, right? Or we have a disruption of normal gene function. So in this case now, what happens if we inject the gene or the gene is injected into the wrong area? We're gonna have insertional mutagenesis or a mutation via insertion, and we'll have disruption of all sorts of genetic functioning. So off target effects. So remember that we stated before, and it's kind of difficult to get therapeutic genes where they need to go, right? So if they actually go off track, they can cause plenty of issues in specific cells, right? The successes now. Let's look at the pros now, right? Of gene therapy. There are recent advancements now. Despite the challenges, there have been some significant advancements of gene therapy, especially for cystic fibrosis, right? Researchers have developed more efficient viral vectors, such as the adeno-associated viruses, AAVs, with which have some promising results delivering therapeutic genes. So now we're looking at the fact no, right, that we actually have more research going on, right, and we're actually increasing the effectiveness of our viral vectors in actually inputting our therapeutic genes, right? Several clinical trials have been conducted to evaluate the gene therapy approaches for cystic fibrosis. Some of these trials have demonstrated improvement in lung function, reduction in pulmonary exacer- um, exacerbations rather, and other positive outcomes for safe patients receiving gene therapy, right? So what we're looking at, through clinical trials, right, via gene therapy, right, we can begin to actually learn a little bit more about how the genes really affect the body and how we can increase the ways of, increase the effectiveness of gene therapy for general diseases, right? Why I'm using cystic fibrosis, yet again, is because that is one of the things, right, that we look at, right? The case study of cystic fibrosis is what the syllabus asks us to look at, all right? Precision medicine approaches, medicine approaches now, right? Advancements in precision medicine have enabled the development of personalized gene therapy. So now gene therapy, unlike other areas of treatment where it's general, gene therapy can be specifically tailored to the individual. So that's a pro, right? That's a success there, right? So it can be tailored to the individual based on their specific gene mutations. This approach holds great potential for improving the efficacy and safety of gene therapy, right? So for CF and other genetic disorders. So generally, for gene therapy, that specificity to the specific individual, so tailoring it to exactly what the individual themselves need, right? That is something that could increase efficiency, right, efficacy and safety, right, of gene therapy on a whole. And one last one now. Well, another one, the combination therapies. Researchers are exploring combination therapies that combine gene therapy with other treatments and modalities, such as small molecule drugs or gene editing technologies like CRISPR-Cas9, to enhance the therapeutic benefits and address multiple aspects of safe pathology. So what we're generally looking at is that we can actually combine gene therapy with other things. If we look at regular internal medicine, many drugs can interact with other drugs that will actually create a lot of issues, right? But with gene therapy now, we can actually combine the therapies, right, more or less safer without it actually disrupting anything else in the body. So we have that there as a success. All right. So that's really it for the gene therapy. So all I did was list out 4 successes and 4 challenges, right? So the syllabus asks us in 4.3 of module 2 to discuss the successes and challenges of gene therapy in modern medicine, and I discussed 4 challenges and 4 successes. All right? And now we move on to 4.4, the last part of aspects of genetic engineering, which is the implications of GMOs. All right? Genetically modified organisms. So the implications on humans. No, right? Remember this objective 4.4 asks us to look at the implications of the use of genetically modified organisms on humans and the environment. So here we have humans, and the next slide is the environment. So for humans, we have nutritional benefits. For anybody who does food and nutrition, I did food and nutrition in CAPE. For anybody who did it, right, we would have extensively looked at GMOs. GMO is a huge part of the food and nutrition unit 1 syllabus, right? So food and nutrition students would have looked at these and already had this in the bag. right? So if you had that understanding from Unit 1 foods, you can use that the same understanding here, right? So nutritional benefits here would be very important. We usually enrich our fortified foods by genetically modifying them. Note, almost every single food that we consume today has already been genetically modified. All of them, majority of them, all of them have been genetically modified over the years. You cannot escape genetically modified foods. Just cannot happen, right? So what we generally look at now is when the GMOs are engineered, they can we can engineer foods that actually have more nutritional value, right? Has more vitamins, minerals, and stuff like that. We fortify them with new nutrients, right? We enrich them with new vitamins, and it can actually be better to improve the public health outcomes in particularly developing countries, right? So if you look at the epidemic in Asia, what was happening, the increase in night blindness, right, with the deficiency in vitamin A. If we increase the amount of vitamin A, we can have stuff like golden rice. That is an example of a genetically modified organism, right, that can help or benefit, right, society. So I think you guys should go out and look at different genetically modified organisms that benefit humans, right? Through nutritional benefits. So you have multiple other ones that we could look at, right? Another implication could be the allergenicity and health risks. One concern regarding GMOs is the potential for the unintended allergenicity or other adverse health effects. Before GMOs are approved for human consumptions, rigorous safety assessments are conducted to evaluate the potential risks to human health. However, some individuals may still have concerns about long-term effects of consuming GMOs. So we don't actually know generally the complete long-term effects of consuming GMOs, but at the current period within human history, it seems just about fine. But if there are long-term effects, we'll figure that out further along, right? But generally, the GMOs, right, can actually cause some allergenicity within humans. So some people could be allergic to the modifications that arise, right? So you can actually become allergic to some GMOs, right? So we have that there. Reduce pesticide use. Again, we could fortify the plants to become much stronger and fortify them against any type of disease. So in the agricultural industry, right, we can engineer crops to be resistant to pests and other diseases, reducing the need for chemical pesticides. Right, this can lead to decreased pesticides exposure for farmers and consumers, right, and it can decrease the risk of eutrophication, right? Eutrophication, you look at in seed biology, will decrease the risk of using those artificial stuff that will now lead to soil pollution and water pollution, right? To reduce the risk of those pesticide related health problems, right? But we'll also have the ethical and societal considerations. The use of GMOs specifically raises an ethical and societal question related to food sovereignty, biodiversity, and the corporate control over the food supply. Some people may have concerns over the concentration of power in the hands of few multinational corporations that dominate the GMO industry. So some corporations completely have control over what we eat and what happens to our food and how they change the food and give us to eat, right? So some persons have issues with that. Some people are wondering whether or not, right, these pose huge ethical and societal challenges and religious issues, right, as it relates to humans now increasing the benefit, right, of these crops, of these foods, right? And the religious implications and societal implications usually comes in when we talk about creating humans or genetically modifying humans, right? Because we can genetically modify humans in which babies can be born that are immune to cancer, that are immune to many diseases. We can create humans that are immune. We can genetically modify a human. Let's say that you want your child to be 7 feet. We can make that happen. You want your child to have blue eyes? Done. We want your child to be genetically predisposed to be muscular? Done. We can do that. But there are ethical and religious and societal concerns about doing those things and genetically modifying certain organisms. For plants, it is bad and not so bad. But when you start modifying humans, right, and animals, then well, humans and other animals, because humans are just other animals, when we start modifying humans and other species, right, then it becomes a slight concern for the religious community. For me specifically, science itself, right, if there is some use of genetic modifications that will better, right, well, let me not better society, right, but where the pros outweigh the cons, right, I think that tests and trials should be run, but that's a personal thing, you know. Some people think say not supposed to touch at all, that you're not supposed to genetically modify anything. That's how some people believe, but it's really a belief system, right? And the science speaks for itself. So I think that more research needs to be done so we can figure out whether or not there are actual real implications where the cons outweigh the pros. So that's what I think there. All right. So that is that. Implications on the environment now, the impact on biodiversity. GMOs may have unintended effects on biodiversity, right, through gene flow and hybridization of with wild or non-GMO products. This can potentially lead to genetic contamination of wild populations and loss of biodiversity, particularly in regions where GMOs are widely cultivated. So when I genetically modify my organisms, right? If they go out and start reproducing with other organisms in the environment, we don't know the repercussions that can have on the biodiversity. We don't know. We don't know if my genetically modified organism, right? If like let's say that my genetically modified potato, right, now goes through sexual reproduction with another potato, a wild potato. What will that do? We don't know what that will do. There's research done that we can actually know through in science. We actually know what that can do depending on what we modified it to be, but it can affect the biodiversity of the populations. It can lead to biodiversity loss, right? As the modified organism, listen now carefully, right? As the modified organism will be more resistant to diseases, etc., etc. Therefore, it will now take, oh, if a disease comes through, right, it will actually destroy the organisms that have the different variations that are susceptible to the disease, but our biodiversity will be lowered because our GMO crops will be way more what would say now resistant, way more resilient, right? Therefore, we could have an issue where the GMO crops took over the wider population, right? And then we have a loss of biodiversity as all the GMO crops are the same. So they are no longer biodiverse. It's just one crop going across. And that's what happened for farmland, right? When you look at farmland, when you look at acres in North America, where there's just miles upon miles of just wheat, miles upon miles of just corn, right? It reduces the biodiversity. Biology Unit 2 students will look at that as well. Herbicide resistance and weed management. Some GMOs are engineered to be resistant to herbicides such as glycophosphates, right? While this can lead to more efficient weed control in the short term, over reliance on herb-resistant GMOs may contribute to the development of herb-resistant weeds, leading to increased use of herbicides over time. So if we keep on using these herbicides on the weeds, right, the weeds themselves, right, may become resistant, right, and that's because of that entire relationship there, right. Non-target effects as well. GMO engineered for pest resistance or other traits are may inadvertently harm non-target organisms, such as beneficial insects or soil microbes. This can disrupt the ecological process and can cascade effects on ecosystem health. Right? So we have that there. So what we're saying here now, is that if our organisms themselves affect insect populations, right? Insects are consumed by something else, and that is consumed by something else, and there's an entire food chain. So what happens when we disrupt the food chains and unravel the food chains? It can affect the strength and resilience of the ecosystems, right? So that's another effect there. So what we could do, what happens here, what we're saying here, is that if we genetically modify an organism, right, to be pest resistant, right? What will the pest eat? It's no pest resistant. Now you actually cut off areas within food chains and not affecting the overall balance of the ecosystem. Right? Now we have contamination and gene flow. So GMO crops can cross pollinate with related species and varieties, leading to genetic contamination of non GMO crops or wild relatives, right? So contamination events have raised concerns among organic farmers and proponents of GMO-free agriculture, as well as potential trade disruptions due to differing regulations on GMOs in different countries. So GMO genes can now leach into non-GMO products and now lead to a concern, right, within the agricultural community, right? And note that there are different regulations for regular products that different from GMO products, right? So we have stuff like that, right? So there are different regulations for that. So we have that there. And that is really where we come to a close. Right? So that was what we're looking at for gene therapy and the implications of genetically modified organisms on humans and on the environment. And that brings the entire topic, aspects of genetic engineering, to a close.